Now let's go right to the fundamentals. How was that for you guys? Tough. Yeah, some interesting concepts, isn't there?
Yeah, I don't even know. I didn't even
Did it still went a little over the head?
Yeah.
Well, let's try to bring it down to earth. If we can. Did you like the videos put into the quiz?
That was nice.
That was kind of cool, right?
Yeah, it just seemed like your questions got harder, especially when you want us to actually put in words. It was in the words.
Yeah, that is.
And it's very
I
challenging.
know.
It's good because you make us think more and learn more. But
Well, as you can tell, I'm into it. On this chapter, I'm into it.
put it, if I ain't gonna lie, I didn't like it. But I get why you did it.
So let's see. We first start with this chapter here. The electricity chapter. All right, so let's go through a little bit of physiology of the nervous system, and then we go through a little bit of fundamental anatomy there. Together with the endocrine system, the nervous system controls and coordinates what goes on within all of our body cells. As we learned, the endocrine secrete hormones into the bloodstream from where they find a specific receptor on a target cell. Remember that? And we just did that moment. Those then prompt the cell and act specific ways. The nervous system works fundamentally similar the same way, except that nerve cells don't secrete substances into the bloodstream, but rather send an electrical signal along cellular extensions, which are cable-like structures that we commonly call nerves. Have you ever hit your funny bone? That's a nerve. That's what those extensions are. Lots of them together make a nerve. And so they go and bring that signal directly to the target cell, where from there then a neurotransmitter is secreted, which is the chemical. Neurotransmitter is a chemical like a hormone. It's just a different name because it's in a different system. And then that is picked up by a specific receptor, which makes the next cell do a specific action. So that's very similar in these two functions, except the endocrine system uses the bloodstream to distribute its messages, and the nervous system uses cables like telephone cables to send its messages. What do I want from you? The nervous system fundamentally consists of two parts: the cables, the nerves, and then they get the electrical impulse between the body and the brain, and then the brain itself, which integrates all the information that we receive. Can you name what we call those two distinct parts? So the peripheral nervous system is what? The brain or the cables? The cables. That's out in the periphery. And the central is the brain and also the spinal cord. With that, let's look at that picture here. So we got the brain and the spinal cord, that's the central nervous system, and then whatever comes out from them, we call that the peripheral nervous system. I think this stuff is really cool. In order for the brain to control our cells functioning, it has to be informed what's going on both internally as well as outside of our bodies. So internally, that's like, gee, my stomach hurts, or what, something like that. Or the chemical levels of something is a little off, or that kind of stuff. The outside is, what do you feel, what do you see, what do you hear, what do you get touch with, temperature, all that kind of stuff. So their nerves carry informative messages into the brain, some of them do, while others send commands. So once we get the information come in to the brain or the spinal cord, the brain and the spinal cord will process it. Like, let's say you go from a dark, let's say, I don't know, the simple one is a reflex. The simple one is you touch something hot. The information going into the brain is like, oh, it's hot temperature. The brain does some thinking, what are we going to do about that? And it sends the information back out to the muscles going like, well, we're going to pull away because it's hot. We don't want to get burned. And so you have information going out, and you got information coming in, and then you got information going back out. What impulses are carried in nerves, carrying them to the brain? From the body to the brain. Sensory. We sense. So you think that you're sensing something. You're sensing some light. You're sensing some touch. You're sensing some pain. And so that's the sensory nervous system brings the information in, and then in the brain spinal cord we integrate it, and then the motor sends the information back out. And that's basically our negative feedback loop that we have. We got the receptor detects a change and sends a sensory impulse to the brain or core. So that's like we get picked, oops, goes in, and then for interpretation. And then after that integration part, then the information motor impulse sent to a effector. And here that's a muscle that then retracts the finger from the painful stimuli. Ooh, look at that. Now we're going to get straight to electricity. Where is it? Where is it? The endocrine system controls body functions that are ongoing and take time, such as growth, the cell's uptake of nutrients, for example. Sending hormones to the bloodstream is perfect for that. On the other hand, the nervous system controls body cell functions that require a quick response, such as moving the arms to turn the steering wheel in a car in order to avoid an accident. To send these signals with maximum speed, the body makes use of the concept of electricity. Have you ever put your hand in a socket? That's electricity. Which is generated when negative charges travel through wires. So it's a negative positive question. In the body, what molecules do we use to generate electricity? What molecules do we have that are negative or positive? Ions. Ions are negative or positive because they either, when they make a chemical bond, one of the atoms either takes an electron or gives an electron. They don't share an electron. That's why they are positive or negative. So they're not covalently bonded. All right, that's good. Magnets either have a positive or negative side. The positive side is attracted to the negative of another magnet. Do you know when you put a magnet on a fridge, you know both sides stick, right? I have a really cute video of my granddaughter, and she figures it out. She's like, why doesn't it stick? And then she turns it around. That was the last time she had to worry about it. It's very cute. I mean, the brain is so plastic when it's like that young. It's like one-time exposure, it's learned. I know, please, can we bring it back to that? Well, only the positive experiences, of course. Okay, so when we have the negative and the positive go to one another, they snap. We got the two positive sides, they repel each other. Charges work the same way. If allowed to flow freely, would you expect positive charges or positive charged molecules to move towards a positive or a negative, a positively charged field? So positive charges, would you expect them to move to a positively charged field? False, right? You wouldn't expect that. You want it to be repelled. Expect to be repelled. Let's see where we see that. So that's the ion stuff here, right? And then here is the likes repel. That's the positive to positive. Opposites attract. So that's the concept that we need to understand. If you got a negative thing, it wants to go to the positive thing. If you got a positive thing, it wants to go to the negative thing. A boundary separates charged fields. In the body, charges are carried on chemical molecules that have weight and mass. If they cannot move freely from one side to another, we can push them actively to one side and then use that established gradient to create a nerve impulse or electrical surge within the body. What cell in the structure creates the boundary that in a cable, it's the plastic around the cable. For us, it's the phospholipid bilayer. It's the cell membrane. So the phospholipid bilayer is a fat layer. That's the plastic. So positive and negative are hydrophilic. They can't cross. They leak a little bit because nothing's perfect in there, but generally speaking, they stay on one side of the boundary or the other side of the boundary. The only way we can move them across boundaries is when we have channels. So here's the cell membrane. It's an oil film. Positive negatives cannot cross the oil film unless we have channels in it that can cross it. We have gates that can open and close. So that's cool because we can use that for a nerve impulse. Since the molecules cannot cross this boundary, the body can use ion pumps to actively move specific ions to one side of the boundary. This way, the body sets up a steep chemical gradient for ions in nerves. So the sodium ions are pumped to the outside and the potassium to the inside. Did you watch that? Did that help? Good. When a nerve impulse is elicited, specific channels in the plasma membrane open, allowing a specific type of ions to cross the membrane freely, which is passively. All right, so what the pumps do, where's a pump? Give me a pump. There's a pump. The pumps, they actively take 3 sodiums, pump them out, and take 2 potassiums and pump them in. And then once they're in that place, we got the cell membrane as the boundary that lets them from traveling across. They cannot cross that. And so what that does, it makes a lot of potassiums on the inside of the cell and a lot of sodiums on the outside of the cell. And that's a gradient that is established. And so let's see. Will the ions, so when we have these gradients established, so a lot of potassium on the outside of the cell, let's go right there. If we have a channel here, and we open a channel here that lets the sodium go to one side or the other, which side do you think they will travel if they can freely travel? Down here, right? Down the concentration gradient. It's like you're top of merit, you go down with a bike, you don't have to pedal. Going up, you have to pedal. That's a concentration gradient, a steep gradient in a hill. And so this is great because in a nerve, what we can do with this, we can take an active process that takes time, going up the hill, it's slow and deliberate. We can do that when we're just sitting around. And we can make this gradient. So when we need a fast process to happen, we can then just open a channel, open a gate, and something passively can happen, which passive you can associate with, in that respect, is quick. It's much quicker to shift all those sodiums to the inside of the cell if it's passive than shipping them to one side or the other if you have to ship them with active processes that take ATP. And so a nerve impulse has to be very, very fast. And so therefore, we're going to use this passive channel concept to do the nerve impulse, and we use the pumps to establish the situation that we can create a nerve impulse. Does that make sense? Good. I hope so. Because it's getting a little bit more complicated. Not too much, just a little bit. So this was down, right? Down the concentration gradient. This is complicated, but you can go back to the cell chapter with this one a little bit. So now let's move along into another concept, and that's voltage. Voltage is the difference in charge between 2 fields. In our body's discussion, that means it's a difference in charge in how much electricity, how much negativity, how much positivity do we have between just inside and just outside of the cell membrane. If a specific ion channel opens and these little charged molecules then cross the membrane, so we're gonna have a charge is a difference between positive and negative between these 2 fields. And the 2 fields are like just outside the cell membrane, just outside of the cell membrane, just inside of the cell membrane. So we now, so if we then have these channels and they open, these little charged molecules cross the membrane, that negativity or positivity changes a little bit because we take all these positive ions to one side and to the other side. So in a nerve impulse, what happens is we have all these sodiums established on the outside and all these potassiums right on the inside of the cell membrane along the nerve. And when the channel opens, when we make a channel here again, these are all positive charged things. They then all fluctuate in here, and they bring more positive to the inside. And so that changes the voltage. That changes how many positives and how many negatives we got around. And the voltage change, we can detect that. We can measure that. And the nerve can measure that too. That's the nerve, that's how the nerve impulse basically happens, with that shift of that electricity, of that charge. And so let's keep going with that. Does that make sense?
So like sending it?
Yes. So when we look at the action potential, that's where we're going to go with that. How it's going to travel along an nerve, basically. This whole discussion is when you hit your funny bone and you feel it going down to the pinky, how does that happen? How is it going to travel from there down to the pinky? Or, you know, basically. All the funky thing is the fact you feel like you hit the pinky happens, and the noticing of that happening in the brain, right? We're going to get to that. Come on. Oh, there it is. So, where was I? Detectable changes in a body are, so these movement of these electricity things these movement of these charges is the electricity. Body uses specific amounts of changes in voltage as triggers to create nerve impulses that are sent along nerves through the body. This is wild, right? Of course. To do this, the body's excitable cells need to have an established voltage while at rest. It's called the resting membrane potential. What structure makes this resting membrane potential possible? Sodium potassium pump. We talked a bunch about it already. That sucker right here. So a resting membrane potential means in our nerves, in all those cables, before we hit the funny bone cables, right along the nerve on the inside and the outside of the cell membrane of that nerve, we have a given amount of positivities and negativities. And that given amount is called the resting membrane potential. It's not, the nerve is not firing nothing, it's at rest. And that is negative 70 millivolts. It's like nothing. It's very, you can't really feel it, but it's electricity. And it's a little bit more negative on the inside. So it's slightly more negative as a whole, all molecules put together on the inside as compared to the outside. So now right there, so we have this established. And so right there now, when we want to fire a nerve, we hit that funny bone, a channel will open that is a sodium channel. So it lets all this positive from the outside flow in, making the inside more positive. Changing this voltage from negative 70 to positive something. So it flips that. And then, so that's when you fire a nerve. And then a little bit later, another channel opens, and that other channel codes for potassium. And you've got all these potassiums on the inside. Now, the funny thing is, which we always get confused in that, the potassiums are also positive. But they're going to be able to carry, as a molecule, they carry that positive charge back to the outside, bringing it back to the resting membrane potential. Except now, the chemicals are all mixed up. And so now the sodium potassium pump goes to work while the nerve is not firing and reestablishes this chemical situation. And so we call that an electrochemical event. The electricity is the positive and the negative, and that's carried on the chemical, which is in this situation is the sodium or the potassium. Good. You got that?
So is that kind of like negative feedback forward? Like it's getting its...
Yeah, I mean it's negative. Yeah, because you bring it out of bounds to bring it back into bounds. You know, it's like it's from that end. Yeah. But we don't describe it as such.
Right.
Mm-hmm. So that's the pump. So let's see what else is here in the text. If you hit the funny bone, we got that. the pinky, the pressure is what we call an ulnar nerve elicited to fire. That is what a nerve impulse is. Or also, a nerve impulse is an action potential as well, technically speaking. What happens is that when the threshold is strong enough, sodium channels, when the threshold is when you hit the funny bone hard enough and it wants to fire, that's the threshold. Sodium channels in the cell wall open and let a gazillions of these positive charges into the cell. That makes the inside of the cell's charge voltage a little bit more positive. Or we can also say that less negative, of course. That's kind of the way this science stuff goes. The voltage changes, and as a result, we feel a little shock when we hit that phony bone. This portion of the entire nerve impulse is called the depolarization. So that is named because the voltage or the polarity across the cell membrane is decreased. So we go from, let's say, we have a resting membrane potential of negative 70 millivolts, we go to positive 20 millivolts. That's depolarization. This becomes less. Goes actually to the flips to the positive. The other portion of the nerve impulse is called repolarization. Its goal is to send all those positively charged molecules, charges back to the outside of the cell in order to re-establish the original resting membrane potential as far as voltage is concerned. The chemicals are messed up. The voltage is proper. Which ion does do that? The K, potassium. K is potassium. You got that, right? K is potassium. N is sodium. Na.
Now I know.
Oh, I know. It's one of those, yeah, it's the German influence, you know. Sodium is natrium in German, I know. The advantage I have is speaking German, I guess, right? So let's talk about that a little bit more in here, because that is really the kahuna. That's the kahuna. We just did the kahuna. The nerve impulse is the kahuna. We talked about all that. There it is. So this is the visual, this graph. I like this graph. I gotta make it bigger. I know. This here is voltage. And this here is time. And voltage is that here, this is negative 70. This is resting membrane potential. That's why this is right there. That's before we fire that first piece. When we fire, we want to fire, sodium channels open. That's that Na, sodium. So here, sodium channel open, sodium goes into the cell. That's what happens, right? Sodium enters the cell. That changes the voltage from negative 70 to about positive 20, they say, más o menos. That's depolarization. Then at that point, sodium channels close their voltage gate. When the voltage shifts the right way, they close them or they open them. And potassium channels open. Potassium fluctuates out of the cell. So we got the K flowing out of the cell. That is right here. Sodium channels close. Oh, no, here. Potassium channels open, potassium exits the cell, restoring negative voltage. Then the voltage, when going up, it goes right back down because all those positive charges go right back out of the cell, making it back negative. Except we make it a little extra negative because the potassium channels close slowly, and that overshoots it a little. We call that hyperpolarization. And that ends up having to do with the nerve not being able to fire for a given moment, and that helps timing it, how the nerve firing pattern can go. So that's called repolarization, and then all of the channels are closed at that point, and the sodium-potassium pump reestablishes the resting membrane potential in terms of the chemical mix-up. You got that? No?
Yes.
Maybe a little bit?
You're gonna study it.
And then you go look at the test review, And then hopefully, then if you don't understand the test tube, then you come talk to me. Or maybe talk to some other people first. You don't have to, you can just straight talk to me too. But make sure you understand for sure what's on the test review. Because some of this might take a couple of times. You know, but the factors are, you're dealing with charges crossing a cell membrane, and you're dealing with chemicals crossing the cell membrane. Right? Which charges, which chemicals cross when determines of what the electricity is across the cell membrane. And that's basically it. Easy said. So the interesting thing about an action potential is they're all equally strong. So that means, for example, if I hear a sound, one impulse from the ear will communicate to the brain that I heard a sound, but it doesn't tell it anything about its intensity, such as loudness or of the sound. How do neurons code for intensity of an impulse? Frequency. So, rapid firing one after the other makes for a stronger stimulus. It's just a coding question. Just a coding question. A nerve impulse is an electrical wave of opening and closing sodium channels along the length of the nerve. It moves forward because when the voltage in one section changes, sodium channels of the next segments open. Even though this process is fast, it can be sped up with a wonderful trick. Since ions are hydrophilic, they do not cross the fat barrier. A fat barrier. The body can now wrap phospholipid bilayer sheets around the nerves. At those sections, ions cannot exchange, and the impulse has to jump to the next section with no wrapping. What are those phospholipid bilayer sheets called? Ta-da! Myelin sheet. So the myelin is the insulator of nerves. If a kid, and I found this video of myself learning how to walk. And my dad sent me some DVD that they transferred the Super 8 onto. And I gotta clip it out because it's actually really funny. You know, the kid walks and like first they fall over. And then they stand back up. But then they take 2 steps and they fall over. And then they stand. And then 3 and 4 and 5. And as soon as, next morning they can walk. That shows you how fast the nerve gets insulated with myelin, because that's when the myelin grows. So those nerves first are slow. That's why they're looking like they're drunk. And then the nerves are needed to fire faster, to not fall over, and they build that myelin, and the nerves grows fast. And so that's the way it grows. And then the negative of that is the pathology is multiple sclerosis. It decreases the myelin sheet. And that's MS. And then it depends on how bad it is, but that's what the pathologies of that. So it's sort of the learning walking in reverse thing. So that's a cool thing, and then so you have to the impulse has to go from these little segments where there's nothing to the next little segments where there's nothing, and they call those the nodes of Ranvier, where we have the depolarization happening, and that greatly speeds up a nerve impulse. The other thing that speeds up a nerve impulse is how thick the nerve is. Very skinny nerves are not as fast, the thicker nerves are faster. So the pain nerves are very slow and ongoing, and the reaction balancing nerves are thick. Now that we know how the fundamentals of how nerves electrical impulses works, let's move to the anatomy. First, we concern ourselves with studying the small single cells within the nervous system, then move to study the brain and the spinal cord And some receptors like what's in the eye, what's in the ear, and that kind of stuff. Two main types, oh yeah, it's very interesting. Two main types are, cell types are the main worker bees in the system. Neurons are the ones that send electrical nerve impulses around the body. That's what we just learned. And then they actually also receive stimuli messages from other neurons and then process them. What the heck did I want to say with that sentence? Oh yeah, so they send the stimulus, they receive stimulus from other nerves and from the outside of the body, or the inside, but from the outside of the brain, spinal cord, and then they process the information that we received. The other set of cell types that we have, they help and support the neurons. That means they're giving them nutrients or defend against intruders, for example. They also insulate cells, which help them speed up the nerve impulse, which is what we just learned. What do we call the helper cells? Huh? These? The glia cells. Glia, neuroglia. Glia means glue. So they hold the nervous system together. So let's see, that goes to the next chapter. And that's right here. There we go. Glia cells. Neuroglia. Glia means glue. Good. So when we look at neurons, that's the ones we really talk about in this chapter mostly. The actual doing cells do what the system is intended to do. In terms of, they come in many shapes. We have small ones, we have large ones, we have ones with many extensions, some with just a few extensions. But generically speaking, when we talk about a neuron, we look at three main portions of a neuron. So we got a soma, we got a dendrite, and we got an axon. And so what is what? So what is the soma? The who? Metabolic center is the soma. The dendrite is what? Receiving or sending? Dendrite. Receiving is dendrite. And axon? Sending.
They send a receptor. The receptors, or they send a receptor
to the next receptor organ. Yeah, send a nerve impulse to the next receptor organ. So let's look at it on here. There we go. That's a generic nerve looking thing. So that centerpiece where the nucleus is, that's the cell body, or also known as soma or pericardium. I put all the AKs I can find in here so you have cross-referencing. The dendrite is branches that come off of the cell body, up to 1,000 branches. Look at that, looks very interesting here. So this is the stuff that can grow when you study, when it hurts a little in the brain. That's when this grows. I know. At least then you know it's doing something good. Well, hopefully if you focus on the good stuff. Those are dendrites, those are they receive stimuli from other neurons. And so what actually happens is a lot of other neurons attached to this neuron within the brain. Most neurons are neurons that attach to other neurons. And all these other neurons talk into this neuron. And they talk into this neuron by saying yes or no, by voting. Meaning fire or not fire. It's a binary, it's a computer system. And then the stimuli, they all sort of want to travel to this place right here that is the beginning phase of this next thing that's called the axon, which is the sending extension. And when all these stimuli, these voices saying, hey, fire or don't fire, positive or negative kind, depending on how loud those voices get, at this area here, which is called the axon hillock, which is written somewhere. Then that nerve decides to fire, and then we create an action potential that goes down the axon to the extension, and at the extension we release a chemical which is a neurotransmitter that goes to the next nerve, to a muscle, or to a gland. So that after a nerve, all we have is neurons to go to a neuron, to a muscle, or to a gland. I think that's very important because that makes it finite. And so, so all these, when I look at what's happening, where was that? Oh, that was in physiology still. So when we look at an action potential, that way that we describe it, that's really this nerve impulse here that gets elicited at this place. When we look at all the extensions that feed from information from other nerves into it, we're not looking at all action potential, we're looking at, they call them graded potentials. They're like all voices that then need to accumulate as volts. How many volts do I get? And when there's enough volts, the voltage changes sufficiently and then this fires. And then that's an all or non-firing event, we call that. The only way you can stop is by cutting it. But you've got to be really precise knowing when to cut it. Because it's fast. Nerve impulse is fast. Good, so that sets that clear. That's anatomy. In order to describe different type of neuron, we consider extensions coming off the cell body. The most numerous type of neuron is mostly found in the brain and the spinal cord. Has many, that's the one we just looked at. It has a body with many extensions that are called, many extensions coming out of the body to the dendrite side and then one to the axon side. And we call that neuron type multipolar because it has many things coming off the cell body. Then we have some neurons within the cell, but within the cell's body placed with a long extension only, bipolar neurons. So we have, those look like this. We have the nucleus, one extension on one side, one extension on the other side. One extension receives the stimulus, the other one sends it. So one dendrite and one axon. Those are called bipolar, bi meaning two, because they only have two extensions. We find that style in the eyes. Very few places we find that, but it's the one we have to describe. Then lastly, we also have neurons that are all our sensory neurons. They are carrying the information from the body through the spinal cord to the brain. So, what happens is, a neuron has a cell body and then the extensions. The cell body is very vulnerable to being injured because that's a metabolic center. That's what the nucleus is itself. That's the genetic material. So we got to be, not a bit careful with that. The extensions, we can squeeze that. That's all right. We can hit the funny bone. It doesn't die. It just shoots it down, and then we'll do it again if you hit it again. So in order for being protected, well, all the nerves that send information out from the brain to the body, all those cell bodies are going to be in the brain and in the spinal cord. Also the cells the neurons that travel within the nervous system, all those cell bodies are very well protected in the skull and the brain and the spinal cord, the skull and the spine itself. But the cell bodies of the cells that send information in, they're lying outside of that bony encased structure, and they're very vulnerable. So what they have made, and what evolution has created, I guess, is they figured out we can take all, on one, when you look at the spinal cord, all the places where the vertebrae is, there's these nerves coming in and out on each segment. And so what they decided, all the nerves that come in, let's just put all the cell bodies in a cluster together close to the spinal cord and be protected. Strength in numbers. So that's what they did. So we started out with these sensory cells. These are sensory cells, these bipolars, but we cannot have these nuclei right in the middle. What they did is they made one long extension and then have another side extension and then a nucleus up there. And then another nerve comes in parallel, because there's many nerves coming in, and they all have in this side extension, and all those nuclei are all in the same cluster, well protected. Strength in numbers is kind of the theme here. And so those, what are those cell type called? Pseudo-unipolar.
Yeah, that's what I thought it was, but it was wrong.
Was it wrong?
It's a multipolar.
No, you have to put like that whole.
Well, then I made a mistake. So I'll type it down. But
I put that there before it's on
Yeah.
yeah, no, I'll check it out. But pseudo-unipolar is what I'm looking for.
Yeah.
That makes sense. I do read through these things, you know, I'm completely, well, it's 6 in the morning, so who knows, right? Which question was that? What number? Huh? 13?
Yeah, number 13.
Okay, good. So multipolar, most numerous one, bipolar with 2 extensions, pseudo-unipolar, only 1 extension. That's the visual of it. Good. And I'm doing all that and then the answer's wrong, huh? I haven't checked that. To a large extent, neural processing depends on counting up and interpreting all of the receiving stimuli. The chemicals released at the end of the nerves, the cables, are either excitatory or inhibitory to the receiving neuron. If all excitatory messages combined are stronger than inhibitory ones, the neuron will fire its own nerve impulse. That's what I just described with all the dendrites coming in, right? And then we go along that cable. At its end, the impulse stimulates the release of its own chemical, which is a neurotransmitter. The NT then actually floats around in a narrow space to find its way to a corresponding receptor on the recipient of the message, which is the next neuron, a muscle, or a gland. What do we call this area where the neurotransmission takes place? Synapse. Did I get that right?
Yeah. Yes.
Good. Isn't that great? You know, you can turn it around on me. And you get credit anyway. There we go. So the nerve fires down the action potential, like here to an X nerve, and at that section there, that's what this is. The electricity comes down and it stimulates the release of vesicles which have little chemicals in it, the neurotransmitters, that travel across the space onto receptors which either excite the next situation or inhibit it. Either excite it or inhibit it. It's always this binary thing. So we have the synapse of when we want to look at the whole components of it, we have a presynaptic area, that's the end of the neuron that sends it, a synaptic cleft, a gap, and then a postsynaptic membrane, which is on the recipient. And the recipient is a muscle, a nerve, or a gland. And you heard of these things? GABA, glutamate, maybe not acetylcholine.
Absolutely not.
Acetylcholine goes to all of the muscles, skeletal muscles. You heard of GABA though, right? For nerve problems, they give you GABA. GABA is some good stuff. Yeah. Yeah, you should try that out. No. You got to talk to your doctor about that. I know, right? Neural connections between neurons can be formed or destroyed depending on their usage. Haha. This is the key component of this whole chapter. Neuronal pathways are most significant for nervous system function. The more a pathway is stimulated, the stronger it becomes. This is why I encourage retrieval exercise for learning the stuff, you know, the flashcards for labeling exercise, blah, 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 blah. I know. It's the same broken record. It also means that if we always complain how bad the world is, for example, we're more likely to experience depressive moods. On the other hand, if we stop and smell the flowers, so to speak, I should put a TED talk in here, we encourage the gratitude pathways and fire it up and get stronger, which most likely makes us more happy. Neurons that fire together wire together. Strength in the connections are strengthened, the groove gets stronger. The strengthening of specific neuronal pathways happens when receiving neurons grow extensions back to the neuron that stimulated them. Awesome, right? I think so. What do we call that type of neural circuit? Oops, it should go up. It's a little delayed here. It's down below. Reverberating. Why isn't it going on? There we go, reverberating circuit. So let's look at that. So that's simple electricity understanding a little bit. It's this one. You know, you have to learn a muscle in the arm. It's a pain in the butt. The test is tomorrow. Shit, I'm excited about it. No, nervous, anxious. But I'm going to do it anyway. I'm going to sit down and study it. And I'm going down and I'm reading the muscle. Then I make a flashcard that gets stronger, right? The making of the flashcard gets stronger. And then I have to think of the name of the muscle. And at some point, the last nerve feeds back to the previous neurons and the pathway stimulates itself. And that's when it gets nice and strong, and that's when we have neuroplasticity happen, which means neurons that fire together, wire together. And so you make a path, a groove in the brain, a connection, and if you do a lot of it, or you make one first, and if you do a lot of it, it gets stronger and stronger and stronger. And if you do less of that, if you don't have these thoughts or whatever, it gets weaker and weaker and weaker. I know. So if we look at everything like an experiment, which is one of the neuroplastic tools they try to tell chronic pain patients, for example, oh, I can't do this because it hurts when I do this. Oh, and the brain feeds itself. Think about the brain works. Oh, we'll talk about that when we get to the homunculus in the brain. I mean, there's areas in the brain here when you body sensations, like the arm stuff that happens in the arm goes to one specific place here that codes for the hand or the arm, for all the inputs going in. And so when I have a pain in my hand, for example, the pain centers light up here where the hand is. If that pain stays there for a long time, this area in the brain becomes not as clear anymore. Get confused a little bit over time. So then my pain moves from the hand to the arm also. And so that's how we can then have pain in the arm without having a problem in the arm. Because the brain is not understanding what's going on. It's overstimulated with pain stuff. And so we can, and that's chronic pain. A lot of times that's chronic pain. Or at least one part of chronic pain. I'm not saying the pain is in the brain. That's false thinking. But there is a component of the pain that's not necessarily from the physiological damage to the body tissues. And so what we can do with that is we can then work in some way with the neuroplasticity, because a lot of times it hurts. I can't move it if it hurts. Well, if I don't move it less, it will hurt more because the brain gets more and more confused. So starting to use the arm again and move it again will redefine these areas better, and the pain will decrease over time. And so that's why when you look at that, oh, let's do the experiment. It hurt when I do it before, but maybe I just have to do it a little bit and go with it and do an experiment, and then it fixes it and it works, or it helps. That's what that means. And I think that's, it's a good outlook.
Yeah.
Shit, man, my foot hurt. I had an injury in the summer on my foot. When I get up and work, it's like, oh man, it hurts. Then I take my 3-5 mile hike or walk. It doesn't no pain in the foot. But if I don't do it, if I start complaining in the morning, I keep complaining because it hurts, then it keeps hurting. I learned that trick from my mom. She's 75. She's like, yeah, my feet hurt. I have to walk so they don't hurt no more. That don't show that on TV. Which is the American addiction. Come on. This is the way it is. Soon it's all over the world. Okay, enough of that talk, huh? A couple more concepts. When we look at the brain in particular, it's a very sensitive organ that needs to be well protected. Oh yeah. It does this by being enclosed within the skull, a hard helmet-like bone. In addition, a thin layer of watery liquid surrounds the brain, protecting it further by absorbing shock, making it practically weightless. Did you ever swim in a pool? Try to run a dash in a pool. It's real hard, man. It's like, oh man, I'm like on the moon. It doesn't come back down when I push up real good. And so that's that weightless thing. So the brain is in a weightless environment. The problem with the brain being enclosed comes when it gets swollen. The skull provides a finite space that doesn't allow for much expansion. Infection is the biggest threat for that, besides shock trauma. Shock trauma is when we get hit over the head pretty hard. That's like a concussion. But that other, the infection, is pretty bad. Since infections travel mostly by the bloodstream, the brain's blood capillaries have processes from neural support cells that cling to them and seal them off so that the type of substances entering into the brain tissue is limited. What do we call that thing? Blood-brain barrier.
That's what I would put it.
Oh yeah, that was the other one. Did I do that wrong too? That was number 16, huh? Will you put that? You all put that? Well, you all get a point.
Well, you probably have to put a barrier part.
I did that too, and it's maybe like a hyphen.
Well, maybe I misspelled it. You know, that could happen because I have to lay all the answers behind it. We'll look it up. When you do lab, I'll look it up. But I got it down. You all get the point.
Thanks.
I know. So let's go to that real quick. See, the good news is now you remember that because you were all like, "What's wrong with him?" Now that one you remember. So that's that. So you got these glial cells are called astrocytes. So you got these astrocytes clinging. They have these podocyte type structures around them. Well, I guess they don't call them podocytes, but they cling around the capillaries to make sure only stuff that goes into the brain is what we want to go into the brain. Good. And then last but not least, which part of the neuron can regenerate? Did I get that right?
17.
Oh, look at that. I want to be finished. I get too many wrong.
This one's wrong too.
Oh, that's why I'm jumping over it. When we look at nerves in our body, the ulnar nerve, for example, which gets excited when we hit the funny bone, most of them have cables. This is a bad sentence. I'll work on that. That sends sensory information from the body to the brain, as well as the one sent impulse from the motor back from the brain to the body. What do we call a nerve that carries both sensory and motor impulses? Yeah. I got it wrong?
Yeah.
What was it?
16.
17.
17. Oh boy.
I think mine was right.
Well, you know, there is like when I edit this stuff, when I open it to edit the questions, the ones these, sometimes it only gives me like I have 5 things written, it gives me one is correct. So I have to open it, edit it, and then save it, and then it shows all right. So maybe there is something behind the software that's still a little goofy.
So we'll get
So I'm gonna, yeah, you get that right? Yeah, of course. Of course. Yeah, if you know you're right, you get it right.
Okay.
If you know I'm wrong, you get it right. So that way, I gotta make sure you're thinking you're right, trick you a little bit. Yeah, it's all done purposely.
Anxiety and.
Oh, no. Well, then you text me. If the anxiety goes too high, you text me, and I can say, "Oh, we'll take care of it." Or you just have that mantra in my head. Anyway, which part of the neuron can regenerate? The axon. Did I get that right?
Yeah, you did.
Ah. One.
Yeah. I put that instead of wrong.
You what?
I put that instead of wrong. I always get it wrong.
Well, I'm going to review that
Well,
one
I
too.
put a capital
Just
A,
for
and it was
semantics.
wrong, and then when I went back and put a small a, they made it correct.
No, but I put a capital and a small, and you both speak.
Well, it was right when I took
Anyway, what I wanted to do now is... Oh, wait, we also have the other questions, right? How was the other stuff?
What stuff? Yeah, the question.
Yeah, the concepts.
What concept?
Yeah, the ones after the test.
Explain the mechanism, the action, the AP, and the reverberating circuit. You got those?
No.
Not everybody does it.
I haven't done it yet.
The neuroplasticity.
Well, then do that. Even if you get laid, do it. You wanna explain the acting potential in your own words. You wanna work that in your head through a couple times. That's all I ask for on that one. That opening, closing sodium potassium channel thing.
Okay.
That one, because if you understand that, you have a lot of, then you can add on stuff later. But that's one of those like muscle contraction type things. You just need to know about it. And then explain the neural reverberating circuit. So we just explained it. And how it influences the brain. How does neuroplasticity fundamentally work? Well, I just went over it. I'm going to put that video up. And then, you know, we'll have this answer for next week. If you haven't done it yet, you can review the video and gather the information out of that. I'm going to put the integration video up that I'm taking right now. Yeah. Good.